I want to start with my talk. Focus on plants from high elevations in the Bromeliad family. Um, so why bromeliads from high elevation? So a couple years ago, I moved to Colombia, where most people were studying plants from high elevations, and it's an exciting place because I don't know if you've seen this paper from Marignan. It looks like evolution is happening really rapidly and also with a very adaptive signature. So I got excited on that topic and started reading a lot of literature, like stuff in lupines and speletias, like showing a very fascinating case of adaptive radiation in those high mountains. But at the same time, you read papers like this showing that those ecosystems in the high elevation Andes are quickly disappearing for climate change, for human intervention. So they need attention. We need to understand what's happening in those high mountains. And in the case of bromeliads, well, there are some interesting cases to study. So we have our high elevation lineages, so it's cool to start studying them in depth. So because I'm a systematist, our approach to understand these biodiversity crisis and these interesting issues is first, go to the field a lot, go to the herbaria, study the species, understand where the interesting questions are. So I spend a lot of time doing taxonomy, describing new species, exploring. Then with those questions, move to the more modern tools and use phylogenetics molecules to answer those questions. And then perhaps use that information to inform people that are concerned about how to protect these plants. So myself, I participate a lot in these red list projects. And then when we go to these meetings, we go back with more questions that we can probably solve with the results that we can produce in our studies. So my two study systems are the two big genera that radiated mainly in the high elevation Andes. One is the genus Greigia and the genus Puya, which are in this phylogeny from Gifnish, are these radiations that occur mainly in the Andean mountains. So the first project that we started with my colleague Rachel and Alejandro Zuluaga started with Puya. So as I told you, I wanted to know more about this genus, know about the taxonomy. If you don't know this genus, you can recognize it for these winged seeds, these spiral corollas after anthesis, and capsules. And this is the most famous Puya, the queen of the Andes, they call her, Puya raimondi. And these are some very cool species from Chile that have these sterile tips, Puya chilensis. So I started with the taxonomic work a long time ago, very inspired by Rachel, who conducted a lot of field work before me. And then I followed her steps, also went to the field a lot, collected many species across the Andes. Rachel collected around here, I collected a lot around here. Also visited botanical gardens, went to many herbaria to see the specimens. And we are now getting to the end of having a very nice taxonomic treatment of this group. Next. Is it working? Oh, back. Well, just to give you a little overview of what's the diversity like in this group, it has more or less 250 species and many more to discover. Here are some examples of some of the new species we've discovered. Despite being very showy, giant plants, people don't collect them because they're very spiny. They're really hard to collect. You need to bring a machete to get, and like a lot of bags, because one individual can fill 3 bags. So we're discovering a lot of new species, not because they're rare, it's because people don't collect. Well, the taxonomic work is coming soon, but what has been already published is this work on phylogenetics of Puya, the phylogenomics of Puya that came out this week on Monday, I think. So you can already go to Google Scholar, take a look. This was a collaborative work with Rachel, Alejandro Zuluaga, Julio Betancourt, and Emma Stone-Smith. And I'm gonna present you some of the results. To give you a little bit of context, well, Puya, you know, is mainly in the Andes. There are some species that get a little outside of the Andean Cordillera. Those are species that mainly live on rocks, near rocks. But predominantly, they live in the Puna and Páramo, which are the high elevation ecosystems towards the central Andes and the northern Andes. And they're also very diverse in coastal Chile and also in the inter-Andean valleys in Peru, Argentina, and Bolivia. This group is also morphologically very diverse. You can see big contrasts of species in terms of size. We have this 10 centimeter Puya in Colombia, in Venezuela, and we have the giant Puya raimondi, which can be up to 10 meters. We also have a lot of different architectures in inflorescences, from very congested to very lax and simple inflorescences. Many types of flowers, a lot of flower variation, probably many pollination systems, many strategies for protecting the flowers, either from the freeze or from herbivores, and also cool strategies on life form. So we have semelparous plants, and we also have, semelparous plants and heteroparous plants in the genus. So our approach was to use the angiosperm 353. We took a risk there. And we used the supercontics of the 353 bait set and carried out a phylogenomics analysis with coalescent models. But then we tried to have time-calibrated tree for Puya using the sortadate, which is strategy to select a few of the 353 genes just to do the BIS analysis for dating the trees. So we selected after the we recovered 344 trees. It worked very well for us. And then we selected 25 genes to use this dating process and then all the biogeography after that. And we also wanted to understand what were, in a very simple exploration, if there were any traits that were important for the plants to move from the valleys and the lowlands to the high elevation ecosystems, to see if there are some interesting adaptations to those environments. So this is the main tree that we got. It's a lot of information there, but I'm just gonna point out some interesting features here. First, these blue pies indicate that the origin of the genus was in Chile. That was already known from previous work in bromeliads. And from there, apparently the plants move north towards the Huancabamba Depression. And from there, they enter the Cordilleras and colonize the deep valleys of Peru, mainly in Peru and then later Bolivia. After that colonization, there is another return to Chile with the Chilean yellow puyas. This is a very interesting case. What we still need to explore better, because these Chilean yellow puyas and these Chilean blue puyas, despite being separated for so many millions of years, they look identical. They just differ in color of the flower. So there's probably some interesting hybridization case here that we didn't explore, but we have the data to continue working on this. And then we have a big colonization of these F and G zones, which are the northern Andes. We have a fully monophyletic group in the northern Andes, which is very exciting for other research because we have a full lineage that can teach us about evolution in these high elevation ecosystems of the northern Andes, which are the Páramos and the high valleys. Um, just in general, the colonization started in interandean valleys, and then it moved to this high elevation, which we call the sky islands, and then different events of diversification occurred there. In our trait analysis, we also, well, before I talk about that, here's a trend that you can see. These bars indicate the range of elevation of each of the species in the phylogeny. And you can see how these clades down here have big ranges on elevation, but only in the northern Andean clades, they became almost exclusively high elevation taxa. Then we selected these 10 traits and performed this correlation test with the Pagel test, and then a new method that was published by Liam Revel recently, last year, to see correlation of the between the movement to the lowlands to the highlands to like freezing conditions, and if it was related to any of these 10 traits, which we coded in a binary way. So we tested photosynthesis, we tested size, we tested life history, indument in the leaves, a lot of inflorescence, architecture traits, protection of flowers by floral bracts, spines or loss of spines in the bracts, and indument around the flowers. And we only found correlation with life history, floral imbrication, visibility of the main rachis, and floral protection and lanite indument around the flowers. Just to give you a quick overview of how those events happen in the evolution of the group. Here on the left, you can see the reconstruction of the arrivals to high elevation. So high elevation are in black and low elevation are in gray. And here are the traits that we evaluated. So this is semel parity. As you can see, it only happened in high elevation. This is an adaptation of life already in high elevation ecosystems. It was not present in low elevation taxa. So we conclude that transitions to life history traits tend to occur more in sky islands and then get lost in high valleys. This is for the exposition of the main rachis of the inflorescence. You can see that the protection of the main rachis or the exposed tissue is more common in high elevation taxa and is predominant in low elevation taxa, that the inflorescences are lax and with their axis very exposed. Similarly, in the racemes, the flowers tend to be more imbricate in high elevation taxa and more lax in low elevation taxa. We assume this is a way to protect from freezing the soft tissue, mainly. And we also saw that the floral bracts tend to be more protective of the flowers in high elevation taxa than in low elevation taxa, where the species of Puya tend to have more pollinators, more floral variability than in the Páramos. So our guess is they need to show their flowers fully in order to get pollinated in lowlands, and this is a harder job to do in high elevation taxa. Okay, so with that, we are basically completing a lot of the approach with Puya. All this work made us famous in the conservation community, so they call us to help with the assessments of the IUCN to see how likely that these species are to go extinct. And right now we have all the high elevation taxa of Puya, all the endemic species already categorized for extinction risk. And this has been used this is work in progress. There is this new methodology of the IUCN of proposing key biodiversity areas based on species that have been already categorized as inputs to propose new areas for conservation. It's just not for high elevation taxa, but for many taxa that have been categorized. And because we have to work with communities too, well, we also produce some information to the people to understand why these plants are important in the páramos, why protecting their habitats rather than the species is more relevant in those areas. And just very quickly, the new project I am carrying out right now is centered in this very problematic genus Greigia. It's clearly monophyletic, I think. There are no phylogenies of this group. And it's problematic because the plants are very conservative in their morphology. They all look alike. They tend to live together. The species are very sympatric. And the floral characters are really hard to study because the inflorescences are very compressed and they basically look the same across all the species. So it's a challenging group for taxonomy, but this is always my first step, try to approach the taxonomy, do a lot of exploration, try to see the plants in the wild, explore new characters that are not traditionally used in the older monographs, because even Lyman Smith said this is the hardest group he had to study. So a lot of pictures in the field will help. And one of my students is currently helping me a lot with the Colombian ones. We already have a taxonomic treatment for the ones in Colombia. A lot of new species are coming from this genus, which was basically mostly wrong in all the herbaria that we want, badly identified. And I also have another student, Santiago Cirosi, trying to understand what pollinates these plants, because it's another mystery of science. These plants have lateral inflorescences. They're always close to the ground, and they have these white petals, flowers that smell like fermented milk. We were able to see some insects arriving, like these braconid wasps. We know the bear eats these fruits a lot. This is the poop of the spectacle bear, full of seeds of Greigia and fruits. So we're trying to understand the floral biology of this group. We have some data on phenology, some data on the cycles of the flowers, but this is still work in progress. And I also currently, 2 years ago, I joined the National Science Foundation grant by Laura Lagomarcin and her collaborators to study the modeled effect of biogeography and trade evolution in the diversification of the high elevation Andes. And I convinced them that Greigia was a very good group, a very good example to understand those processes in a different way than the other lineages that they chose. So we are currently generating a lot of genomic data for Greigias. We expect to collect more samples from countries outside of Colombia, which is where I collected a lot. And some exciting results will be presented in 5 years or before. Okay, that's it. Thank you to everybody that has collaborated with me. I don't do this on my own. And thank you to you.
Thank you very much. Now we'll have time for 1 or 2 questions.
So really a great achievement by the team to get this Puya phylogeny. I wanted to suggest a hypothesis regarding these very massive inflorescences. We know in Espalicia, which grows in the Páramo and experiences winter every night, summer every day, that they're really facing a problem with water in the morning because of the frozen ground, and so they store it in the pith of the stem, and experiments have shown that having insulation around the pith is vital to prevent it from freezing and dying. And I guess I would suggest for the high elevation Puyas that they're investing huge amounts of water every day when they're in flower, producing all that nectar, basically, and that insulating that peduncle, or the inflorescence stem, would be vital. But of course, the you want to show two things. One is that, in fact, it's acting as a capacitor, that it's storing the water outside of the soil. And secondly, that it is killed if you remove some of the insulation, remove some of the flowers. Anyway, that might be something exciting really to explore in the future.
Yeah, these are traits that we need to test. These hypotheses we need to test in the field with specific work.
Any other questions?
Thanks for the talk. Excellent. I was curious about the booklet that you showed about Puya that you took to the communities, and I would like to hear how do people local react when you explain the importance of the taxonomic work?
Well, that little booklet was just, it's not released yet. The communities don't know the book yet. We work with the communities to create it with another colleague from Bogota. She's the one that's really spending time with workshops and teaching people. This is part of my students' master thesis. She's trying to tell people that things living in sympatry are important, and Puyas in the Páramos are living in sympatry, and that understanding where those places, where those species are located together are very important to protect rather than the species. So she's trying to teach them what a niche means, what a sympatry means, what are the evolutionary processes that are happening right now. So they can go to the field and when they see different puyas of different sizes, that's a key area to protect and to be paying attention what animals come, etc., etc. But they haven't seen the little booklet. We're still working on it, and hopefully I can go to workshops. Yep.
Thank you again. And if you have any other questions, you can ask the presenter at the lunch break.